So in Bio 311C, one of the first things that you learn about are all the different functional groups that you'll find on the large biomolecules that we'll cover later. With these functional groups, you need to keep three things in mind: their polarity, their ability to hydrogen bond, which these two are related in their ability to allow the molecule to interact with water, and the pH, how acidic or basic the functional group will make the molecule. So the first one that we're going to cover is the hydroxyl group. This is just an oxygen and a hydrogen. The electronegative oxygen pulls on electrons from the hydrogen, making this polar. Since there's an extremely electronegative atom attached to a hydrogen, this does allow for hydrogen bonding to occur. However, since the oxygen isn't quite strong enough to pull on the electron so that the hydrogen snaps off, which would make this an acid, that's not the case. So this is neither acidic or basic. Alright. So the next one that you're likely to see is the carbonyl group. This is simply a double bonded oxygen to one of the carbons in your skeleton. So this can occur in two forms. It can occur in the middle of the carbon skeleton like this, which makes it a ketone. I like to think of a ketone as the oxygen's inside. So a key goes inside of a lock, so here it is, ketone, lock in key. However, if it occurs that, find that hydrogen, if the carbonyl group is on the end of the molecule, like this, and this is an aldehyde. Aldehyde's a little bit harder, but I like to think of the carbonyl group trying to hide on the end, so it's going to hide, but it's in a horrible position. Kind of weird, but oh well. Because this oxygen is pulling on the electrons, it is polar. But since the extremely electronegative oxygen is not attached to a hydrogen, it can't participate in hydrogen bonding. And since there is really no hydrogen to pop off, it does not have an acidity or basicity. Okay. So the next one that we'll cover is the carboxyl group, which is sort of like a carbonyl group and a hydroxyl group that have combined. So let's put a methyl on it. So looking at this right here, it looks like we have the hydroxyl group right here, and it looks like we have the carbonyl group right here. But since they're combined on this singular carbon, it tends to act a little bit differently. So what's happening is this super electronegative oxygen is pulling on electrons this way, and this oxygen is also pulling on electrons from the carbon. However, since this is double bonded, it's going to be a little bit closer to the carbon, and it's actually going to start pulling electrons through the oxygen. So even though that this oxygen's all the way on the other side, it's pulling electrons in a way that really weakens this bond between the oxygen and the hydrogen. So because of that, the hydrogen tends to more easily fall off. And so this hydrogen ion is just a proton, which makes this an acid. And as you likely have guessed, since you have a super electronegative oxygen on either side, polarity is involved. And since this hydrogen is connected to this oxygen, there's also hydrogen bonding. Nice. So this is our primarily acidic functional group. So the next one that we tend to cover is the amino group, which this one's a little wonky right now. But the amino group consists of a nitrogen attached to three different hydrogens. So this one is wonky because it is a base. And it's a base in that it can absorb a hydrogen at will. So you can find it at NH2 like this. But then if there's a floating proton in the environment, it can absorb it and become NH3. So by absorbing the proton from the environment, it makes the solution around it more basic. So the amino group is basic. And since nitrogen is fairly electronegative, not as electronegative as oxygen, but electronegative nonetheless, it is causing polarity, and it is allowing for hydrogen bonding. Okay, So, the sulfhydryl group is just a sulfur attached to a hydrogen. It's very similar to the hydroxyl group, but instead of an oxygen, it's a sulfur. So it tends to have pretty similar reactivity, except you'll see this in some special amino acids such as cysteine, which allows for disulfide bridges to form. So just a pretty cool one to keep in mind. So then we have the phosphate group, which is kind of large and clunky. So this one's sort of unique in that where it attaches to the carbon skeleton is over here on this oxygen. So you have a phosphate double bound to an oxygen, single bound to what appears to be a hydroxyl, another hydroxyl, and then an oxygen that attaches to the carbon skeleton. And so this is large and clunky and kind of full of energy, as we'll see with ATP, sort of. But it has oxygens that are pulling on the phosphate, hydrogens attached to those oxygens. So we have both polarity, hydrogen bonding, but no change in pH. Finally, we have the methyl group, which is not really a functional group because it lacks the qualities of the others. So you have a central carbon attached to 3 hydrogens. Carbon and hydrogen have similar electronegativities, so they form nonpolar bonds. And because of this, this entire thing is nonpolar. So that's what makes it different from all the rest. All of the functional groups that we've covered so far have been polar, except for the methyl group. As such, it also cannot participate in hydrogen bonding. And since these bonds are pretty equal to begin with, there's no way that it's going to start dropping hydrogens to make the surrounding solution acidic. So that's what makes the methyl group special. We cover it because it's sometimes found on DNA to change the transcription of different genes. And so with that, those are all the functional groups. If you have a set like this at home, you can feel free to make these on your own, but hopefully this video has sufficed for those of you that don't.